Good morning, everyone, and welcome. I'm pleased to be here with you today. We're about to delve into an important topic, technology transfer for sterile products. My name is Dr. S.K. Singh, and I hold a PhD in this field. Over the course of this presentation, we'll explore key aspects of transferring technology for sterile manufacturing, including best practices, challenges, and solutions. So, let's get started and look forward to a productive session together. Let's take a closer look at the definition of technology transfer. Essentially, it is a systematic process where we transfer product, process, and knowledge from one site to another. This isn't just about moving things around. It's about ensuring that the receiving site can accurately reproduce the product, follow the process correctly, and understand the knowledge involved. The key goals here are reproducibility, compliance, and maintaining product quality. Reproducibility means that the receiving unit, or RU, can consistently produce the same quality of product each time. Compliance ensures that all regulatory and internal standards are met throughout the transfer process. And of course, maintaining product quality is central to the entire effort. When we talk about success in technology transfer, what really matters is that the receiving unit can reliably replicate the product. That's the ultimate goal, making sure that what they produce matches what was originally intended, both in quality and consistency. This systematic transfer process is critical for scaling up production, expanding to new sites, or even ensuring that a product remains consistent across different manufacturing locations. Today, we'll be focusing on the objective of understanding technology transfer, or TT, specifically in the context of sterile products. Our aim is to grasp how TT applies across different types of sterile formulations and ensure compliance with current regulatory standards and aseptic requirements. We will cover three main areas: small volume parenterals, or SVPs, which include products like ampules and vials with less than 100 milliliters, large volume parenterals, or LVPs, such as IV fluids exceeding 100 milliliters, and dry powder injections, which are lyophilized or sterile powders. It's important to remember that, according to the World Health Organization, TT isn't limited to just one dosage form. It applies broadly, but sterile products require especially careful attention to detail due to their complexity and the strict standards they must meet. In terms of scope, for SVPs, we're talking about small containers like ampules and vials under 100 ml LVPs, involve larger volume IV fluids. Think bags or bottles over 100 ml, and dry powder injections are lyophilized or sterile powders that need reconstitution before use. Overall, the key takeaway is that technology transfer in sterile products must be meticulously managed to ensure safety, efficacy, and compliance at every step, aligning with global norms and aseptic practices. Now, let's look at the key elements involved in technology transfer. First, there's documentation transfer, which is essential for ensuring that all the process details are accurately shared and understood. Then, process transfer itself focuses on moving the actual manufacturing or analytical processes from one site to another. Analytical method transfer is also critical, as it ensures that testing methods are maintained consistently across locations. Facility and utility qualification are necessary steps to verify that the manufacturing environment and utilities meet the required standards. Training and skill transfer are equally important to make sure the staff is fully equipped to carry out the procedures correctly. Validation ties all these elements together by confirming that the processes are reliable and produce the desired quality. It's important to note that this includes manufacturing, packaging, and analytical procedures, covering the full scope of production. Now, when we talk about sterile technology transfer, it becomes even more critical. Many sterile processes do not involve terminal sterilization, which means the entire process must be meticulously controlled to prevent contamination. Because of this, the contamination risk is significantly higher, making the process more sensitive and demanding. Regulatory agencies focus heavily on aseptic processing, environmental control, and sterility assurance. These are key areas because any lapse can compromise product safety and efficacy. Sterile technology transfer is considered high risk and is subject to strict regulations to ensure patient safety and product quality. So, understanding these elements and their importance helps us appreciate the complexity and the critical nature of sterile process transfers. Now, let's focus on the importance of the sending unit, or SU, in this process. It's crucial that the SU thoroughly assesses the readiness of the receiving unit, or RU, across several key areas. This includes checking the equipment, utilities, and QC systems to ensure everything is prepared for the sterile transfer process. When we look at the sterile transfer process flow, it starts with project initiation and continues through gap analysis, risk assessment using FMEA, transfer protocol development, and then progresses through engineering batches and validation batches before final report and closure. Each of these steps depends on a clear understanding of both sites' capabilities. A critical part of this is the gap analysis, which helps us identify differences between the sending and receiving sites. Recognizing these differences early allows us to address potential issues proactively. Now, roles and responsibilities are key here. The sending unit provides the development data, shares process knowledge, and defines the control strategy. Meanwhile, the receiving unit must ensure facility readiness, confirm aseptic capability, and successfully execute validation activities. Connecting this all together, the overall goal is to ensure a smooth, risk-mitigated transfer. By thoroughly assessing readiness and clearly defining roles, we support the integrity of the sterile process and maintain product quality throughout the transfer. Let's take a closer look at facility and utility considerations. First, the cleanroom classification, which can range from grade A to D, is critical because it determines the level of cleanliness and control needed in the environment. The more sensitive the product, the higher the grade required to ensure sterility and quality. Next, HVAC system validation is essential to maintain the correct temperature, humidity, and air quality. Proper validation ensures that the environment consistently meets the strict standards necessary for sterile manufacturing. Moving on to water systems, WFI, or water for injection, is a key component because it must be ultra-pure to avoid contamination. Similarly, pure steam is used for sterilization processes, so its quality directly impacts product safety. Environmental monitoring also plays a vital role, allowing us to continuously track airborne particles, microorganisms, and other environmental conditions to ensure ongoing compliance. Overall, all these utilities must align precisely with sterile manufacturing requirements. Any mismatch or failure in these systems can compromise product integrity, so meticulous design, validation, and monitoring are absolutely essential throughout the facility. Now, let's take a closer look at some of the key challenges we face in our processes. One major issue is handling large batch sizes, which can complicate consistency and control. Heat sterilization, like autoclaving, is essential, but also tricky because it requires careful management to ensure everything is properly sterilized without damaging the product. Uniformity and endotoxin control are critical as well, because even small deviations can lead to contamination risks or variability in product quality. For example, if the sterilization cycle isn't properly executed, we risk non-sterility, which can compromise the entire batch. Moving on to process transfer, especially for liquid injectables, what we call SVPs, there are critical parameters we need to monitor closely. Filtration using a 0.22 micron filter is vital to remove bacteria and particulates, but if the filter is incompatible with the product, it could lead to bioburden failure. That's why controlling filling accuracy and ensuring the container closure integrity are equally important. They're our last line of defense against contamination. If any of these parameters falter, it can lead to product failure, delays, and increased costs. So, maintaining strict control over these processes is essential to ensure safety, efficacy, and quality across our manufacturing operations. Now, let's talk about the key aspects of analytical method transfer. First, Sterility testing is crucial to ensure our product remains free of microbial contamination. Next, the endotoxin test, often done with the LAL test, helps us confirm that bacterial endotoxins are within safe limits. We also need to assess particulate matter to guarantee the product's clarity and safety. And finally, perform assay and impurity tests to verify potency and purity. A critical point here is that we must ensure the method is reproducible across different sites. Consistency is key to maintaining quality throughout the entire process. Moving on to dry powder injection, there are several critical factors to consider. The lyophilization cycle, for example, must be carefully optimized because an incorrect freeze-drying process can lead to product collapse or instability, which we definitely want to avoid. Residual moisture levels are also vital. Too much moisture can compromise product stability, while too little might cause brittleness. Vial sealing is another important factor. Proper sealing ensures the product remains sterile and stable during storage. To illustrate, an example of an incorrect freeze-drying cycle can result in product collapse or instability, highlighting the importance of precise process control. Lyophilization, in particular, requires tight control of temperature and pressure. Small deviations in these parameters can significantly impact the quality and stability of the final product. Now, when we talk about process transfer in general, it's all about ensuring that these critical parameters, like cycle conditions, moisture levels, and sealing, are consistently controlled across different manufacturing sites. This consistency helps us deliver safe, effective, and high-quality products no matter where they are produced. Let's talk about validation requirements. When we're validating our sterile processes, we typically look at 3 key areas. First, process validation, which usually involves about 3 batches to ensure consistency and reliability. Then, media fill validation, which is critical for confirming that our aseptic techniques are effective without risking actual product. Next, cleaning validation to make sure our sterilization procedures are thorough and effective. And finally, hold time studies, which help us determine how long a product can safely stay in a particular state without compromising its sterility. The most important point here is that all these steps must demonstrate consistent sterility assurance. Nothing is more vital than maintaining that sterility throughout the process. Now, moving on to the challenges we face in sterile transfer and transfer technology. One big issue is scale-up. As we expand our processes, maintaining sterility becomes more complex. Facility differences also pose a challenge because each site may have unique environmental conditions or equipment, which can impact sterility if not properly managed. Environmental variability is another factor. Things like temperature, humidity, or airflow can change from day to day and affect our sterile environments. And of course, process robustness is key. If our processes are robust, meaning they can handle variability without compromising sterility, then transferring them smoothly from development to production is much easier. A robust process essentially means a smoother transfer, reducing the risk of contamination and ensuring consistent product quality. So, overall, focusing on making processes robust is what helps us navigate these challenges effectively. Let's talk about risk management, specifically based on ICH-Q9 guidelines. When it comes to managing risks in sterile transfer technologies, we rely on several tools to help us identify and prioritize potential issues. One of the key tools is FMEA, or Failure Mode and Effects Analysis, which allows us to systematically evaluate possible failure points and their impact. Alongside that, risk ranking helps us prioritize those risks so we can focus our efforts where they matter most. Now, some common risks we encounter include contamination, which can compromise product safety, equipment mismatch, where incompatible tools or systems cause errors, and operator variability, which can introduce inconsistencies in procedures. Recognizing these risks early enables us to implement effective controls. It's important to remember that risk assessment isn't just a checkbox. It's a critical step toward ensuring the success of sterile transfer processes. By thoroughly understanding and managing these risks, We can maintain product integrity and patient safety throughout the manufacturing process. So, to wrap things up, sterile terminal sterilization, or TT, is indeed high risk, but it's absolutely critical to our success in producing safe, effective products. Because of the high stakes involved, it demands strict controls and a deep understanding of the process. We need strong aseptic control at every step to prevent contamination, which means rigorous sterilization procedures, cleanroom practices, and environmental monitoring. Alongside that, having a robust process understanding is essential. Knowing each step inside and out helps us identify potential risks and address them proactively. Regulatory compliance is also a key piece of the puzzle. We must adhere to all relevant standards and guidelines to ensure our processes are validated and our products meet quality expectations. Ultimately, the goal is success, consistent sterile products across all sites. That consistency is what builds trust with our customers and regulators alike. So, by focusing on these critical elements, control, understanding, and compliance, we can achieve reliable, sterile manufacturing that supports our overall mission. For further support, please don't hesitate to reach out to us. We're here to help with any questions or additional information you might need. Remember, your success is important to us, and we want to ensure you feel confident moving forward. Thank you so much for your time and attention today. We appreciate your engagement and look forward to supporting you in your journey.